Our next speaker is Dr. Jen McQuade from MD Anderson. She's a guest speaker. She's going to be speaking I was going to give an update in melanoma, but I think that given the interest of the audience across a lot of oncology disciplines, I was down at MD Anderson recently and heard Jen speak about kind of what diet, microbiome, lifestyle factors are associated with likelihood of benefit to immune therapy. And I thought this was really interesting. And I think this is one of the most common questions we get when we're talking to patients. They all want to say, well, what can I do to try to help increase my chances of benefit? And so I think Jen has done a lot of great research there, and I want to thank her for coming up to Memorial to speak with us today on some of these interesting topics about patients and their lifestyle factors and choices. So thank you, Jen, for coming.
So yeah, thank you, Mike, and the organizers for inviting me here. So as Mike said, today I'm going to be talking about the microbiome and then the interaction with some research that we've been doing with diet, probiotics, and then response to immunotherapy. Just my disclosures. So, the first two talks made me really realize I just finished fellowship 3 years ago, and I'm already completely behind the times in these other malignancies, right? Because this has all just been such a quick change with immune checkpoint inhibitors being approved and really transforming the standard of care in all of these different diseases. So in melanoma, of course, with immunotherapy, our response rates are higher than in other diseases. And, we get some really dramatic and durable responses, especially with a combination of CTLA-4 and PD-1. But even in melanoma, I still have many patients where they just have absolutely zero response whatsoever. And as Mike said, our patients are asking, how do I get into that good bucket, right? Is there something that I can do? And so usually, as we've already, learned when we're thinking about mechanisms of response and resistance, our biomarkers are still really imperfect. So we look at things like, are they T-cell inflamed? What's their PD-L1 expression? We know that there's certain oncogenic signaling pathways that can mediate resistance. But then what about the level of the host? We know that, people of different ages, sex, different obesity statuses, these are all things that actually influence our immune system. How do they influence response to immunotherapy? And then those things you can't necessarily change, but our patients are asking, what about the things that I can change, that there is some data out there about impacting immune response? And then we also got really excited about the idea of the microbiome as kind of being an integrator of all of these different factors, as it is inherently modifiable and influenced by choices that patients can make. Oops. So what is the microbiome? So the microbiome is, of course, these trillions of organisms that live on and inside of us, make up more of our own DNA, have more DNA content than our own human cells. And of course, the largest number of these microbes actually live within our gastrointestinal tract. And prior work has demonstrated that the human microbiome can influence disease prevalence and a lot of inflammatory bowel diseases, but also a lot of autoimmune diseases as well, really pointing to this intimate relationship between the microbiome and immunity. So this is work from the Wargo group at MD Anderson, but also has, of course, been seen in other cohorts as well. So basically that the gut microbiome is associated with response to immune checkpoint blockade. So this was a cohort of patients where the gut microbiome, fecal microbiome specimens were collected, and then looked at the difference between responders to immunotherapy versus non-responders. And what we see in the top left is that responders had a more diverse gut microbiome, meaning more different types of bacteria compared to non-responders. And then there were specific bacteria that were associated with response versus with non-response. And I've highlighted in red some that we were particularly interested in because of prior known associations with diet. And then importantly as well, you actually see that these pro-response bacteria are associated with a more cytolytic T-cell microenvironment. So correlation, not causation, but what was really exciting about this work, and has also been seen by other groups as well, is that you can actually transmit responsiveness to immunotherapy via a fecal microbiota transplant in mice. So if you take poop from a human who responded to immunotherapy or poop from a human who didn't respond, give a fecal microbiota transplant using those specimens into mice, implant a tumor, and then give that mouse immunotherapy. If it got the responder poop, its tumor then responds to immunotherapy. And if you got the non-responder poop, your tumor grows. So an exciting proof of principle, and this has, of course, led to many ongoing clinical trials looking at fecal microbiota transplant in combination with immunotherapy in humans. But again, coming back to this idea of, well, how do you have a good microbiome or a bad microbiome? And what we know is that the gut microbiome is mostly influenced by modifiable factors, so particularly our diet, but also by medications. There's, of course, geographic differences as well, and it's less than 10% kind of genetically determined. So we again know that that diet, coming back to these kind of pro-response bacteria, we noted that a lot of these bacteria are actually bacteria that are that help us digest fiber, and in the process form these short-chain fatty acids, which have previously been implicated in mucosal immunity. So within the MD Anderson cohort, the bacteria identified were, again, these fiber-fermenting bacteria. Interestingly, in the French cohort from Laurent Zitvogel's group, Akkermansia is the dietary associations there look like a very prototypical kind of French diet, rich in coffee, wine, and chocolate. And then a lot of these bacteria that were negatively associated with response are actually bacteria that are enriched in people that consume high amounts of red and processed meats. So we began assessing diet in our patients at the same time that we were collecting microbiome specimens. And what we found is consistent with what we see in other non-cancerous populations. Those patients that were eating a lot of fiber, a lot of fruits and vegetables, a lot of whole grains, had an increased abundance of these pro-response bacteria. And we then looked at the association between dietary fiber intake and response to immunotherapy. And what we see is that our patients that had high baseline dietary fiber intake were almost 5 times more likely to respond to PD-1 immunotherapy. And there was also synergy with the gut microbiome. So the patients that have a type 1 signature, meaning a pro-response gut microbiome signature, and are eating a high fiber diet, 75% of those patients responded to PD-1 blockade. Whereas those with a kind of a anti-response microbiome, low fiber, were the least likely to respond, and then kind of a stepwise progression going up. Again, improvements in progression-free survival as well. So does this make biological sense? It does. So again, these bacteria, when we eat more fiber, we have an increased abundance of these bacteria. They then produce these short-chain fatty acids, and these short-chain fatty acids are actually the primary nutrient source for our gut enterocytes. They help us, the enterocytes then form a nice tight mucosa layer. And then there's been a lot of research between this and looking at mucosal immunity. Now, how this actually then influences anti-tumor immunity, there's still quite a bit of mechanism to be worked out. So then, this is all baseline associations. But again, can we change the microbiome by changing diet? So we've been collaborating with a group at the NCI. And in mice, if you change dietary fiber intake, then you rapidly shift the gut microbiome as well as change response to immunotherapy. Now, how about in humans? So there previously have been controlled feeding studies where they demonstrate that, yes, you can actually rapidly and reliably change the gut microbiome with dramatic changes in fiber intake. So this was a pretty cool study. They actually took African Americans living outside of Pittsburgh and rural Africans in sub-Saharan Africa and switch their diets, right? So you switch from a McDonald's diet to a, kind of agrarian whole grain diet. And when you do that, so this is the short chain fatty acid production, it goes down, 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 and these Africans, as they're eating more processed food, goes up, up, up in the African Americans as they're eating more whole grains. And then we see the opposite with the bacteria that help us metabolize meat. So based on this, we're actually conducting a controlled feeding study at MD Anderson. And so using a bionutrition research core, meaning we will give our patients every calorie-containing nutrient that goes in their mouth over the duration of the study to actually really test, by changing diet, can we change biology? And so we'll take patients that are receiving standard of care anti-PD-1, give them either a high fiber diet, 50 grams of fiber a day coming from diverse sources of whole grains, fruits, and vegetables, or an isocaloric control, otherwise healthy diet. Feed that to them for 12 weeks and then do longitudinal microbiome and immune profiling. But diet change is hard. Wouldn't it be easier if we could just take a pill? And it turns out actually that a lot of our patients are taking probiotics. So again, they've probably read some of the exciting work about the gut microbiome. 42% of our patients are taking probiotics, and those patients actually had a lower diversity of their gut microbiome, were less likely to respond to immunotherapy, and had decreased progression-free survival. Again, maybe they were taking probiotics for other reasons. They were sicker patients. But when we test this in a mouse model, what we find is that if we give mice PD-1 and then give them some over-the-counter probiotics that we just picked up at CVS, then we actually decrease their response to checkpoint blockade. And this has been seen in other populations as well. So again, if you give antibiotic ablation in either humans or mice and then give them probiotics, the probiotics actually decrease the reconstitution of the normal gut microbiota from kind of a competitive exclusion mechanism. So in conclusion, the gut microbiome is distinct between responders and non-responders to immunotherapy. Responsiveness to immunotherapy can be transferred via fecal microbiota transplant in mice, and that approach is also being tested now in humans. Diet and medications are key determinants of the gut microbiome. High dietary fiber intake is associated with improved response, but now we actually need to prospectively test whether increasing dietary fiber can actually change response to immunotherapy. And then we are cautioning our patients against using probiotics outside the context of a clinical trial. And just my acknowledgments, this work is all in collaboration and working with Jen Wargo and Chris Spencer, who's at the Parker Institute here now. Thanks.
Great talk, Jen. This is a really interesting topic. What questions do we have for Jen about this? So I guess I have one real quick one then. Obviously, the microbiota is everywhere in our body, right? Not just in the stool. And there's a lot of attention on stool and stool collection, and there's always inherent challenges with all this. What do we know about oral mucosal microbiota, skin flora, all these other places that bacteria like to live?
Yeah. So in the initial paper in Science, they also looked at the oral microbiome and did not see associations there. The skin microbiome, people are actively collecting and looking at. And then I think the other relevant question is that our from poop samples, we're really not looking at the true gut microbiome, right? So like actually what's in the mucosa as well. So colorectal cancer, they have a beautiful opportunity to actually study that. I think it's pretty hard to get serial colonoscopies in our patients just to look at their microbiome.
We appreciate it enough when they donate their stool, which we are doing, actually. We're collecting that. So just a question here in the back. Wait till the microphone comes. We have a few people online watching, so I want to make sure everyone hears.
Thanks again. I just wanted to ask about the probiotics at the end. Do you think that this isn't going to be a route forward, or are we putting the wrong bacteria in them? Is it just not regulated? If we had put the Clostridiales or one of the other ones, would you expect an effect?
Yeah, so these are, again, unselected kind of over-the-counter probiotics. And what's in those? Well, they're actually the things that are really easy to culture, right? So it's the same thing that's in our fermented foods as well. These nice spore-forming bacteria, again, yes, I do think that that's an interesting way forward for research, and that really, it's about selecting the right bacteria, not just throwing in whatever's easy to culture. Yeah.
Just wondering whether a lot of these people end up taking antibiotics for a brief time, and so how do what's the latest on antibiotics screwing up the diversity and
Yeah, so again, we do know. I mean, there have been a number of studies now showing that that antibiotics lower diversity, and that is associated with decreased response to immunotherapy. Again, is it causative versus correlative? They got antibiotics for a reason. They were sick. So I think what it's led for us to do clinically is just to be more cautious and prudent with our antibiotic use. And what seems to be, at least from the stem cell transplant literature, the most dangerous is those broad spectrum antibiotics, and especially those with anaerobic coverage, because these spore formers are all anaerobes.
Great. Thank you very much, Jen. We'll have some more time during the panel to talk more about this topic.